ABA is a growth inhibitor. Abscisic acid, it is a growth inhibitor. Adicot, isolated abscessin promoting substance from cotton fruits. For that, he gave the term abscessin 1 and abscessin 2. Next, varying isolated dormancy promoting substance from leaves of sycamore, and for the dormancy promoting substance he gave the term dormin. So actually abscessin 2 and dormin both are structurally same. Finally, for the two substance gave the term abscisic acid. Then abscisic acid, rightly 15 carbon sesquiterpenoid, produce both anabolically and also catabolically. Anabolically produced from acetyl coenzyme A and catabolically produced from carotenoids. Then abscisic acid present in almost all plants of the plant kingdom and also present in major parts of the plant. Then physiological effects of auxins, physiological effects of abscisic acid: dormancy, then senescence, then stomatal closure, then perinating buds. So now description: it promotes seed dormancy and bud dormancy. Abscisic acid promotes both seed dormancy and bud dormancy. So seed dormancy is antagonistic property to gibberellins. Actually, gibberellins promote seed germination. Gibberellins promote seed germination, but abscisic acid promotes that dormancy. Then it promotes senescence. Then important point is, when plant facing water stress condition, that the leaves produce abscisic acid. This abscisic acid, it leads to closure of stomata, and thus it helps in reducing transpiration. So it is called antitransparent, and it is natural antitransparent. Then during water stress condition, this hormone is formed and reduce transpiration. So it is also called water stress hormone. And in some aquatic plants, particularly lemna, during summer when water dry up, this ABA produce perinating buds. These perinating buds sink down and reaches to bottom, and again next season, means in rainy season, that the bud germinates. In that way, this lemna plant survive by perinating buds. So these perinating buds are formed by abscisic acid. Simplest phytohormone is ethylene, because It is the only gaseous hormone, and it is structurally similar to carotenoids, because like carotenoids, this ethylene is made up of hydrocarbons, carbon and hydrogen. Then precursor for ethylene is a methionine amino acid. So ethylene is produced from methionine. Importance of methionine: it is initiating amino acid. In any protein synthesis, that the first amino acid is methionine, that methionine is initiating amino acid. Then this ethylene present in almost all parts of the plant. Then Gain suggested that ethylene is considered to be a phytohormone, and the physiological effects of ethylene studied by थिमन and Berg. So थिमन and Berg, they studied that the physiological effects of this ethylene, physiological effects of ethylene. Then ethylene functions. Rightly, ethylene is called fruit ripening hormone. So its main function, it promotes ripening of fruits in banana, grapes, and even tomato. Then next one, triple response growth. Generally, ethylene inhibits longitudinal growth and promotes transverse growth. That is called a triple response growth, reported in pea seedlings. Then ethylene promotes formation of female flowers in a pineapple along with the auxins. Then ethylene also produces adventitious roots. It also produces adventitious roots. Then lateral root formation and also root hair formation for increasing that absorption of water. Then in some plants, inrolling of petals takes place. This inrolling of petals is called sleep disease, inrolling of petals. And in some plants, more growth takes place on upper side of petiole, lower, then lower. So leaf will droop. That condition is called epinasty. Then it promotes senescence, but it breaks dormancy. Then it breaks bud and seed dormancy. In some plants like rice, in rice, that leaf always present above the water. It is due to elongation of petiole. This length of petiole can be increased by that ethylene. Then ethylene is gaseous hormone. There is practical application of ethylene to that plants. Generally, ethylene is applied to plant in the form of ethepon. Then this ethepon release that ethylene. Photoperiodism means relative duration of light in day and night on flowering is called photoperiodism. Photoperiodism was discovered by Garner and Allard in two varieties. One is variety of tobacco called Maryland Mammoth and 𝗂𝗏𝗁𝗀 variety of soybean, 𝗂𝗏𝗁𝗀 variety of soybean. So in 24 hours, that day duration is called a photoperiod, and night duration, night time, is called scotoperiod. Generally, in plants, vegetative mycelium, vegetative meristem converted into reproductive meristem, then into flower. So vegetative meristem converted into reproductive meristem, then into flower. It takes place in presence of light. In presence of light, vegetative meristem converted into reproductive meristem, then flower. This is sequence. If this sequence occur in presence of light, it is called photoperiodism. Or if it occur with the help of temperature, it is called vernalization, and also takes place by the help of phytohormones. Then definite light period is required for conversion of vegetative mycelium, vegetative meristem to reproductive meristem into flower. That definite time period is called critical day length. Generally, this critical day length is 10 to 14 hours. Then, based on critical day length, this plants divided into three types: short day plants, long day plants, and day neutral plants. Short day plants, flowering takes place when they are exposed to only 8 to 10 hours. It means light period is less than critical day length. So more dark period is required. So dark period is crucial. So dark period is continuous. If dark period is interrupted by light, flowering fails. Example for short day plants: tobacco, xanthium, and glycine max. The next one, long day plants. Long day plants, flowering takes place when they are exposed to 14 to 16 hours of light. It means light period is more than critical day length. So light period is crucial. Light period is continuous. If light period is interrupted by dark, that flowering fails. Example for long day plants. Long day plants. Example: henbane, then beetroot and spinach. These are example for long day plants. Day neutral plants, flowering is unaffected by that day or night, called day neutral plants. Example for day neutral plants, cucumber and maize. Vernalization. Vernalization means low temperature treatment given to plant at a seedling stage. So, low temperature treatment given to plant at a seedling stage is called vernalization. And a vernalization was first reported by 𝗧𝗦𝗃𝗀𝗃𝗀. So, vernalization reported by 𝗧𝗦𝗃𝗀𝗃𝗀. Then there are some 𝗧𝗧𝗃𝗃 members. There are some 𝗧𝗧𝗃𝗃 members or graminea members like a wheat. Scientific name is Triticum aestivum, wheat, rye, secale, cereal, barley, hardium, barley scientific name hardium vulgare. These are graminea or 𝗧𝗧𝗃𝗃 members. And actually these are two types of varieties, spring variety and also winter varieties, spring and winter varieties. Then if spring variety sown in spring season, spring variety when sown in spring season, they germinate and harvested at early summer, because spring variety does not need low temperature. Winter variety when sown in spring season, winter variety when sown in winter season, fails to germinate, because in winter season seeds do not germinate. And if sown in spring season, fails to germinate, because in spring season they do not get low temperature. That is why if winter variety sown in autumn season, then flowers are formed and fruits are formed and rightly harvested in summer. Then annuals. In annuals, flowering occurs at the end of first year only, because in annuals that a seed get proper low temperature at a starting stage. But biennials, flowering takes place after passing through winter of second year. Why means in biennials that seeds do not get low temperature at starting stage. So, if biennials, in biennials, if seeds are subjected to low temperature, required low temperature at a starting stage, flowering occurs at the end of first year only. It means actually in biennials, in second year flowering takes place. If proper or required temperature is given to this as biennial seeds at starting stage, that flowering occurs at the end of first year only. And biennials or root crops, example radish and carrot. Next one, seed dormancy. In favorable conditions, if viable seed fails to germinate, it is called a dormancy. In unfavorable conditions, if viable seed fails to germinate, it is called a quiescence. So, first, causes of dormancy. It is generally because of hard seed coat. This hard seed coat, impermeable, hard seed coat, impermeable to oxygen. Example, xanthium. Hard seed coat, impermeable to water. Fabaceae members. Example, trigonella. Then mechanically resist embryo. Mechanically resist embryo. Capsella. It is first one, due to hard seed coat. Then in some plants, immatured embryo, that even at the time of seed germination, the seed contains immatured embryo. In some plants, seed contains fully developed embryo, but seeds fails to germinate because embryo required low temperature. And in some plants, dormancy is due to light. Some plants required light for seed germination, rightly called positively photoblastic seed. Example for positively photoblastic seed, lactuca. Some plants, seed germination is inhibited in presence of light, called negatively photoblastic seed. Example, onion. So, these are causes of dormancy. Then, methods for breaking dormancy. One is scarification. Scarification means removal of hard seed coat. Removal of hard seed coat is called a scarification. This scarification is of two types: mechanical scarification and chemical scarification. Mechanical scarification means rubbing seed against sand. It's called mechanical scarification. Chemical scarification, removal of that seed using that chemicals like sulfuric acid is called a chemical scarification. The next one, stratification. Stratification means subjecting seeds to low temperature is called a stratification. It is also called that a freezing treatment. Then in some Fabaceae members, there is outgrowth which is developed from seed called a strophiole. This strophiole blocks micropyle. That is why entry of water is prevented. Hence dormancy takes place. This trophiolar plugger can be removed by shaking the seeds. Rightly it is called impaction. Actually smallest seed present in orchids, largest seed present in double coconut, largest seed present in double coconut, Lohdecia, double coconut. And actually study of seeds is called the spermatology. And the first seeded plants, gymnosperms. Then structure of seed, simple. Seed means it is surrounded by seed coats. Bitegmic ovule, integments converted into seed coats. So bitegmic ovule, seed contains 2 seed coats. Outer seed coat is called testa. Inner seed coat is called tegmen. Testa is edible in pomegranate. Cotyledons edible in some Fabaceae members. Edible cotyledons. And also anacardium, cashew nut. Then in 𝗂𝗶𝗸 members, only one integument is present. If one integument present means that seed contains single seed coat. In parasitic members, Loranthus, Belenophora, etegmic ovules. So, no seed coats. Seed without seed coat, parasitic members, single seed coat. 𝗂𝗶𝗸 members, 2 seed coats present in dicots and monocots. If 2 seed coats, outer seed coat is called testa, inner seed coat is called tegmen. In most of the dicots, seed is without endosperm. If endosperm is absent, such seeds are called exalbuminous seeds, or also called non-endospermic seeds. Exalbuminous or non-endospermic seeds, common in dicots, but some exceptional. Dicot with endospermic seed, Solanaceae, and dicot with endospermic seed, castor. And in castor, reserve food oils. That is why this castor contains elaioplast. Elaioplast means oil containing leucoplast are called elaioplast. Elaioplast also called oleosomes. Generally, oil seeds contains glyoxysomes. So castor contains elaioplast or oleosomes and glyoxysomes. And in castor, reserve food is oil. So germinating castor seed, RQ value is less than 1. Then in most of the monocots, seeds contains endosperm. So rightly called albuminous seeds or endospermic seeds. Albuminous or endospermic seeds common in monocots. Then some exceptional in monocots, orchids monocots, but seed is non-endospermic. Then point, it is structure of seed. Then seed germination. Actually, seed contains little or less amount of water. So first step during seed germination is imbibition of water. So first step is imbibition of water, seeds imbibe water, then a seed swells, then it develop pressure on seed coats, then seed coats get ruptured, then embryonic axis exposed, and first which structure emit from that seed means radicle, because radicle gives rise to that root system. Afterwards, once this embryo is exposed, then oxygen is available, then aerobic respiration takes place, and that stored food, starch, converted into simple food materials. It means first seed swells, then embryonic axis exposed, then radicle emerges. Once that embryo exposed, oxygen available, so aerobic respiration takes place. This aerobic respiration break down that reserve food into simple form, then mobilization of that food takes place. All these together called seed germination. Then types of seed germination. There are three types of seed germination. One is called epicell. Epicell seed germination means cotyledons are above the ground. If cotyledons above the ground, called epicell. So for example, it is embryonic axis. It is embryonic axis. This embryonic axis contains embryonic node. This embryonic node, it gives cotyledons, then lower side radicle, radicle gives root system, upper side plumule, plumule gives shoot system. So embryonic axis between plumule and embryonic node is called epicotyle. Embryonic axis between cotyledon and radicle is called hypocotyle. So point is types of seed germination, three types. So first one, epicell. Epicell means cotyledons above the ground. Why cotyledons above the ground means this hypocotyle elongates. That is why cotyledons above the ground, called epicell seed germination. Example for epicell seed germination, cucurbita. Then second type of seed germination is hypogel. Hypogel seed germination means cotyledons below the ground. Why cotyledons below the ground means it is due to elongation of epicotyle, called hypogel. Hypogel example, maize and mango. Maize and mango. Then point is third type, viviparous seed germination. Vivipary, it is also called in situ seed germination, or also called aerial seed germination. Aerial seed germination. In situ or vivipary means seed germination takes place inside the fruit. Seed germination occurs inside the fruit, that fruit still attached to parent plant, called vivipary or in situ or aerial germination. It is character of mangroves. Actually mangroves means it shows two characters. One is viviparous germination and second one is that respiratory roots. And example for this rhizophora and Avicennia. Rhizophora and Avicennia. So it is dormancy and germination. So growth is irreversible, permanent increase in size. Actually, almost all parts of plant shows unlimited growth, but limited growth. Which part shows limited growth means leaves. Leaf shows limited growth. Then in plants growth is unique because throughout life growth takes place. Why throughout life growth takes place means because of meristems. Then meristem term was given by Nageli. Then plant growth is called uniform because whenever growth is needed, new cells are added by meristems. So it is called open form of growth. So, plant growth is unique and open form of growth. Unique is unlike other organisms. Open form means whenever growth is needed, new cells added by meristems. Then phases of growth. Phase of growth, three types: meristematic, elongation, maturation zone. Meristematic, it shows slow growth, that is called lag phase. Elongation, it shows rapid growth, called a log phase or exponential. Then that senescent stage, growth is constant. Because of that, S shape or sigmoid curve is obtained. It is exemplified for what means increase in fresh weight or dry weight or number of cells. This is by geometrical application. Then arithmetic growth. In arithmetic growth exemplified by root elongation. And root elongation means it gives what type of graph means that a linear graph. Then next one, conditions. Conditions necessary for growth: water, oxygen, even temperature. Water is essential because water maintains turgidity. Then only in turgid cells, that stretched, cell wall get stretched. Then oxygen is essential for growth because oxygen help in respiration. Respiration gives energy, and in presence of energy, cell division takes place. Then nutrients, both the macro and micro elements, essential for growth because they produce that protoplasm. Then these are conditions for growth. Then next one, plant shows different phases of life cycle, different phases of modifications based on environment. That is called plasticity. Best example for plasticity is heterophyly. Best example is heterophyly. Heterophyly, presence of two kinds of leaves on same plant is called heterophyly. Actually, heterophyly is of three types. One is habitual heterophyly, then environmental heterophyly. Third one, developmental heterophyly. Habitual heterophyly means presence of two kinds of leaves from starting stage onwards. It's called habitual heterophyly. Example for habitual heterophyly: gymnosperm, 𝗧-𝗺𝗃. In 𝗧-𝗃, two types of leaves present from starting stage, rightly scale leaves. Scale leaves are called cataphylls. Another one, normal photosynthetic leaves called foliage leaves. It is for habitual heterophyly. Environmental heterophyly, common in hydrophytes. Best example, ranunculus. Ranunculus is buttercup. Common name is buttercup. In ranunculus, upper leaves are normal and they perform photosynthesis, and the lower leaves are dissected for absorption of water. So it is called environmental heterophyly. Then third one, developmental heterophyly. Developmental heterophyly means changing in structure of leaf from juvenile, means young stage, to mature stage. Is called developmental heterophyly. Example for developmental heterophyly, dolichos. In dolichos, actually young leaf is simple and matured leaf is compound. Even one more example, Parkinsonia is xerophyte. In Parkinsonia, young stage, leaves are bipinnately compound. Bipinnately compound means secondary rachis contains leaflets called bipinetic compound. But after maturity, these leaflets fall off. After maturity, leaflets fall off. The secondary rach is modified into leaf-like and perform the function of photosynthesis. That is called phyllodes. So these are types of heterophyly. Three types: habitual, environmental, and developmental heterophyly. Then some important and extra points for phytohormones: auxins, gibberellins, cytokinins, then abscisic acid and ethylene. See, among these first three, growth promoters, because they promote plant growth, and abscisic acid, growth inhibitor, then ethylene, both growth promoter and also inhibitor. Among these five types, only ethylene is a gaseous hormone and a most simplest hormone. Then nitrogen containing phytohormones, auxins, then cytokinins and ethylene. These are non-nitrogenous containing phytohormones. Non-nitrogenous, gibberellins and ABA. Then mainly auxins and cytokinins, they shows polar movement. Auxins and cytokinins shows polar movement. Polar movement means apex to down or down to apex. Then most common auxin is indolestic acid. Precursor for indolestic acid, tryptophan, and a zinc element essential for tryptophan. Then the main importance of auxins. Auxins act as weedicides. Here both the dicot and monocot weedicides. Dicot weedicide is 2,4-D. Monocot weedicide, that is dalapan. Dalapan means 2,2-dichloropropiononic acid. Propionic acid. Then gibberellins. Here one more important, rooting hormone. Generally rooting hormones, auxins produce roots, but majorly rooting hormone is IBA. It is effective hormone. Then gibberellins, main function of gibberellins, stem elongation. They act on subapical meristems. They act on subapical meristems where internodal differentiation takes place. That is why they help in elongation of internodes. Then these gibberellins are chemically terpenes, and precursor for gibberellins, acetyl coenzyme A. And main function of gibberellins, they promotes seed germination. First reported in barley seed. First reported in barley seed. And first discovered gibberellin is, or gibberellic acid is GA3. Then cytokinins, precursors for cytokinins, purines. But first discovered cytokinin is kinetin. Kinetin is chemically 6-furfuryl aminopurine, but it is not a natural, it is degradable substance. First discovered natural cytokinin is zeatin. First discovered natural cytokine is zeatin. The zeatin was isolated by Latham from immature maize grains along with auxins. Along with auxins, these cytokines help in morphogenesis or organogenesis, formation of plant pods from callus. It's called a morphogenesis or organogenesis. Then employment of living material. Employment of living materials to test biological activities is called bioassay test. Bioassay test for auxins, Avena growth test for auxins. Bioassay test for gibberellins, barley endosperm test. Bioassay test for gibberellins. And a bioassay test for cytokinins, chlorophyll preservation test. Why means this Richmond and Lang found that when detached xanthium leaves treated with cytokinins, that senescence postponed to several days. So such delay of senescence is called Richmond-Lang effect. So that is why for cytokinins bioassay test, chlorophyll preservation test. For gibberellins, barley endosperm test and dwarf maize test. Then ABA is growth inhibitor. ABA, abscisic acid, growth inhibitor. Chemically, these are sesquiterpenoids. Chemically sesquiterpenoids produced from both acetyl coenzyme A and also carotenoids. From acetyl coenzyme A, anabolically it is formed, carotenoids catabolically. The ethylene is gaseous hormone. It is nitrogen containing phytohormone produced from nitrogen containing amino acid. That is what means methionine. And ABA, it is called water stress hormone. ABA is water stress hormone because when plant facing that water stress condition, this ABA is formed and leads to closure of stomata. So it is called water stress hormone and it is natural anti-transparent. Then main function of ethylene. This ethylene help in fruit ripening. Ethylene help in fruit ripening. So it is called fruit ripening hormone. And ethylene treated tobacco leaves, they contains less amount of nicotine. And ethylene promotes flowering even in mango and pineapple. When ethylene is sprayed to that nicotine plant, senescence, synchronous senescence takes place. Synchronous senescence means at a time all leaves become senescent. So it reduces that picking of leaves. So these are important features of that growth hormones. So photoperiodism means duration of day and light on flowering is called photoperiodism. Then photoperiodism discovered by Garner and Allard. And critical day length means a photoperiod required for most of the plants is 10 to 14 hours. It is called a photoperiod or critical day length. Based on critical day length, plants divided into long day, short day day neutral plants. For long day plants, 14 to 16 hours needed. So it is more than critical day length. Short day plants, 8 to 10 hours. It is less than critical day length. And day neutral plants, flowering is unaffected. Right? Then, next, vernalization, reported by 𝗧𝗦𝗃𝗀𝗃. Vernalization, reported by 𝗧𝗦𝗃𝗀. Then, vernalization means low temperature treatment given at a seedling stage is called vernalization. Annuals. In annuals, example for annual cereals. So, cereals lifespan is 1 year. At the end of first year only flowering takes place. Why means? In cereals, that seeds get proper low temperature or required low temperature at starting stage only. That is why at the end of first year only flowering takes place. Biennials. Biennials, starting stage, that seed did not get a proper low temperature. That is why in biennials, flowering takes place only after passing through winter of second year. For a biennial, if proper low temperature is given at a seedling stage, at the end of first year only flowering takes place. It means we can convert biennials into annuals. This statement is wrong. We cannot convert biennials into annuals. Just we can reduce that flowering period from second year to first year. And perennial plants like mango, in perennials, every year, perennials vernalized every year. So, in perennials, flowering takes place every year. Then, dormancy. Dormancy means seed have viable capacity, but they fails to germinate, called dormancy. Then, your extra point is some plants, they require light for germination, called positively photoblastic seeds. Some plants, they do not need light for germination, called negatively photoblastic seeds. Positively and negatively photoblastic seeds. And most common dormancy is due to hard seed coat. And actually removal of hard seed coat is called scarification. Scarification. And stratification. Stratification means low temperature treatment. That low temperature treatment is called stratification. Then particularly in some Fabaceae members, that a dormancy is due to strophiole. Strophiole is outgrowth developed from seed. It blocks micropyle. So it blocks micropyle. So entry of water is prevented. And the strophiole can be removed by simple shaking that seeds, rightly called impaction. So vigorous shaking of seeds is called that impaction. So plant growth is unique because it contains meristems. So that meristems are always divide and they add continuously cells to that plant. Actually, how many types of meristems present? How many types means based on position, 3 types. Apical, intercalary and lateral meristems. Apical and intercalary meristems, they are primary meristems and they help in linear growth. Then lateral meristems present only in gymnosperms and dicots, and later meristems they increases width or that diameter. Then next one, differentiation, de-differentiation and redifferentiation. Redifferentiation. Differentiation, these primary meristems like apical meristems, they gives new cells, that are new cells differentiate to perform that new functions. So differentiation example, primary xylem, primary phloem, parenchyma, collenchyma, sclerenchyma. These are differentiated cells produced from the apical meristems. The next one, de-differentiation. Actually, de-differentiation means some cells lose their power of division, rightly called permanent cells or permanent tissues. These permanent tissues lose their power of division, but some cells regain their power of division and produces that secondary meristems, rightly called de-differentiation. Best example, medullary rays by de-differentiation gives 𝗳𝗏𝗃𝗃 cambium in dicot stem. Then conjunctive tissue and pericycle in dicot root gives both vascular cambium and also that cork cambium. It is de-differentiation. Redifferentiation. Redifferentiation means the secondary meristems gives a secondary tissues called a redifferentiation. Vascular cambium gives secondary xylem and also secondary phloem. Cork cambium, it gives cork and secondary cortex by redifferentiation.